Welcome to Biology 210, Lab 9, Exercise 24, Dilutions. Before you begin, it is suggested that you turn in your lab manual to page 213 through 215 and complete the pre-lab activities. That will help you follow along with what's in this tutorial. In Lab 8 last week, we performed the loop dilution or pour plate method. A loop was used to transfer a mixed culture, which was a broth, and create a cereal dilution. After 48 hours of incubation, the first plate had the most colonies, it was the least dilute, and the third plate had the least number of colonies, it was the most dilute. In this dilution, you used a loop to go from auger deep to auger deep. You then poured the auger deeps into corresponding plates. This week, in exercise 9, we will do a very similar activity, but instead of using auger deeps, we are going to use dilution bottles filled with water, and we're going to use pipettes instead of loops. Lab 8 produced the desired result, but was not quantitative. Quantitative meaning it was not precise. We don't know the exact amount to transfer because a loop was used. We don't know how much exactly is in a loop. In Lab 9, the same idea will be applied, but a pipette, which we do know the exact amount, 1 ml or 0.1 ml, and dilution bottles, which we know the exact amount, which is 99 ml of water, will be used to achieve quantitative results. These are precise amounts. We can then take those amounts and calculate how much is in the original cell concentration or how much in this figure is in this original E. coli test tube. So in this figure, which you'll see in exercise 25, you have a test tube of E. coli. We pipette 1 ml from water bottle A to water bottle B to water bottle C. Each water bottle contains 99 ml of water. We then take varying amounts, 1 ml or 0.1 ml, pipette them into plates. We take the plates, add auger, let them solidify, incubate the plates, and the result is similar to the data in exercise 8. The first plate, 10 to the negative 4, will have lots of colonies, and the last plate, 10 to the negative 7, will have less colonies because it is the most dilute. The purpose of dilutions in microbiology is to reduce the microbial population to a manageable number or size. It is possible in the original test tube, in this case E. coli, there could be billions or trillions of cells, which are too many to count. The result of a cereal dilution is the more dilute, the less colonies. After a cereal dilution is performed, the amount of bacteria in the original sample can be determined. So after we are finished, we can count the colonies on the plates, knowing what the dilution is on the plates, and we can actually calculate how many cells are in the test tube of E. coli. This is important for things like food testing, milk testing, to see if a food sample is safe for consumption. The goals of dilutions is to determine the amount of a bacteria in a sample. We call this the original cell concentration because we want to determine how many bacteria cells are in the original sample. The original cell concentration, or OCC, is the number of bacteria cells in this original sample. So what we can do is take an inoculated, in this case nutrient broth, that is cloudy, indicating lots of bacteria, and dilute it and count the number of colonies on a plate. The sample below is of E. coli, and it is cloudy or turbid, indicating lots of bacteria. We can perform a sear dilution and calculate how many E. coli cells are in the test tube. So how many cells do you think are in this test tube? So what we're going to do in the next set of slides is we're going to go through how you would do the dilutions. You would set them up in water bottles or dilution bottles. You would then add via a pipette various amounts of the dilutions into plates, add auger to the plates so that the cells can grow. The plates would be incubated, and after 48 hours, we would count the number of colonies. We would then do a calculation to figure out how many colonies is going to give us the number of cells in this original test tube. So get your guess ready and we'll see what it looks like at the very end of this tutorial. Exercise 24, dilutions, page 217. So it is helpful for you to follow along in the lab manual as we go. A lot of the information in the next couple of slides is from scientific notation, which we covered at the beginning of the semester. So you should remember or recall that a positive exponent is a whole number. For example, 10 to the 1 is the number 10. That is a whole number. 10 to the 2 is 100. The 2, which is the exponent, represents the number of zeros. A negative exponent is a fraction or a decimal. It is a portion of the original. We will use negative exponents when talking about dilutions. So for example, 10 to the negative 1 is the same as 1 over 10 or 0.1. This is a fraction. 10 to the negative 2 is the same as 1 over 100 or 0.01. Again, this is a negative exponent, therefore a fraction. The exponent, whether it's positive or negative, determines how many places to move the decimal. We move the decimal to the left or to right direction, depending on whether it's a positive or negative exponent. So for example, 3.6 times 10 to the 2 is the correct way to write the number 360 in scientific notation. You place the decimal between the first two whole numbers, which in this case is the 3 and the 6, and you put a 10 to the 2 because you move the decimal 2 places. Another example is 7.24 times 10 to the negative 1. This is a fraction, so we're going to move the decimal to the left and get 0.724. Here are additional resources if you still do not understand scientific notation. You can also look back at the beginning of the lab manual for exercise 2 and 3. Practicing dilutions. So now we're in the lab manual on page 218, and we're going to practice scientific notation. So 0.001. Write your answer on page 218. The first question you should ask yourself is it a negative or positive exponent? The next question is, how many places do you move the 0? 1 times 10 to the negative 2. Number 2, 6,200. Again, positive or negative exponent, and where do you place the decimal point? Since this is a whole number, it's going to have a positive exponent. We place the decimal between the 6 and the 2. The correct answer is 6.2 times 10 to the 3rd. Question 3, 0.415. Write your answer. 0.415, or 4.15, sorry, 4.15 times 10 to the negative 1. It's a negative 1 because it is a decimal or fraction. Question 4, the number 100. Answer, 10 to the 2. There are 2 zeros in 100, and it's a positive exponent. Question 5, 1,000. How many zeros? 3. It is a positive exponent, so 10 to the 3rd. So now that you understand scientific notation, we're going to move to starting to make the dilutions. This is the first part in the process of diluting a nice cloudy sample. So on page 218, there's a summary of all the formulas. These formulas are very important. There are 4 formulas. If you do not understand the formulas as we progress through here, go back and watch the additional resources or contact your instructor for additional questions. It's really important before you get frustrated, because this is a little complicated, that you take the time to practice and review the additional resources. So on the left it says, these formulas are very important. You may be seeing these are a test or a quiz. We will cover each in the next few slides, and you should memorize the formulas and be able to work practice problems. The first formula we're going to discuss is the single dilution bottle. On page 218, I also give you examples of each, but we're going to do those as we progress through the slides. Exercise 24, dilutions, page 219. Diluting samples. So in the picture, you again see that nice cloudy test tube of E. coli. Then on the side, what we're going to get as a result 48 hours later, which are plates with varying amounts of colonies. Remember, this is very similar to what we did a few weeks ago with a loop dilution. The only difference is we use dilution bottles with exact amounts and pipettes with exact amounts. Samples of dense bacterial cultures, foods, or milk are diluted to achieve agar plates with 30 to 300 colonies. If there are less than 30 colonies, then it is too few to count, and we disregard them. They're statistically not valid. If there are more than 300 colonies, then it is too numerous or too many to count, so we disregard those. So why do we want to count colonies on plates? The colonies are used to determine the number of bacteria present in the original sample, so things like food. So if you had a sample of milk and you wanted to know if it was safe to drink, if you had bacteria in it, you would do a dilution, do a plate count, and then count the number of colonies. This can indicate if the food is safe for consumption. So looking at the plates on the screen, plate A, B, C, or D, which of these would actually have between 30 and 300 colonies? If you said plate 3 or plate C, you are correct. Dilutions, page 219. Single dilution bottle. So we start off with one bottle. We're going to take a test tube of E. coli or some other bacteria or sample. We're going to pipette 1 milliliter aseptically into the water bottle. So our formula for one water bottle is the volume added to the bottle divided by the total. So on the picture, on the right-hand side, we see 1 milliliter, that's our volume added, being added to a 99 ml water bottle. So using our formula, it's 1 milliliter divided by the total. 1 plus 99 is 100. So you see below where it says dilution bottle, 1 over 100 or 10 to the negative 2. So 1 over 100 is the same as 10 to the negative 2. Here's an example. 1 milliliter is added to the bottle. We add that to 99 mils in a water bottle. 99 plus 1 is 1 over 100, which is the same as 10 to the negative 2. Again, it's 10 to the negative 2 because there's 2 zeros and it is a fraction. Another example is we could use a 9 ml water bottle. 1 milliliter divided by 99 mils, because that's the volume in the water bottle, plus our 1 ml that we added for our total, so 1 over 10, which is the same as 10 to the negative 1. So we've just done our first dilution of our bacteria. If our bacteria sample was very dense, this would not be enough to get us to a number between 30 and 300. We will continue to dilute further. Exercise 24, dilutions, page 219 through 220. A series of bottles. So as we saw in our initial turbid broth of E. coli, it was very cloudy. And we don't know how many cells are in there. That is the point of this exercise. But it looked very, very cloudy. So if we just diluted it once and we plated it, the likelihood of getting between 30 and 300 colonies is not likely. We probably would have over 300 colonies. So we'll further dilute our solution. It is often necessary to do a series of dilution bottles to achieve the desired dilution, the number of colonies per milliliter. When multiplying numbers with exponents, the exponents are added together. For example, if you have 10 to the negative 2 in the first bottle and 10 to the negative 2 in the second bottle. When we multiply, we add exponents, which makes that 10 to the negative 4. So the first bottle, as you can see in the picture, would be our dilution 10 to the negative 2. We got the 10 to the negative 2 from our dilution bottle of 1 over 100, which was the volume added divided by the total. We did that again for our second bottle, which is 1 over 100, which is another 10 to the negative 2. So when we add those, our second bottle is 10 to the negative 4. So here's another example. We don't have this one listed, but if you literally just added up all of the exponents, 2, 4, 6, 8, our total dilution, if we had 4 dilution bottles with 99 mils of water in each, would be 10 to the negative 8. We'll continue to see this on the next few slides. Here are several additional resources to help you understand. On the second one, stop at the 3 minutes and 30 seconds because the end of that we'll cover at the end of these slides. Next, page 220. Practice setting up a serial dilution. So again, it's a series of bottles. Here you can see we have 3 bottles, each of which is 1 over 100. Remember, each of which is 1 over 100 because it's the volume added, 1 mil, pipetted, divided by the total. 1 plus 99 is 100. So the first bottle is 1 over 100, 10 to the negative 2, same number. The second bottle is 1 over 100. We add those, we're adding exponents, so 10 to the negative 4. Then the third bottle is 1 over 100. So our total would be 1 over 1 million. So 2, 4, 6. So let's do the practice question 6 on page 220. Starting with E. coli, draw, and that's the only way you'll be able to do this, how to make a serial dilution. So it says, hint, how many dilution bottles do you need to achieve a 10 to the negative 6 dilution? So this is the answer, which is our 3 bottles, and we pipetted 1 mil into each of our bottles. So not on this slide and not on these pictures or in your lab manual, but what if you wanted to make a 10 to the negative 8 dilution? How many bottles would you do? So could you use this exact same picture and then just add to it? Correct. You would add 1 more bottle and 1 more milliliter to get 10 to the negative 8. Exercise 24, dilutions, page 221. Plated dilution. So it's fine if the solution is in, or dilution is in the water bottle, but that's not going to give us numbers that we can actually see. So step 2, after we've done our dilution, is to take the dilution and pipette varying amounts into plates. In order to count colonies and determine the amount of bacteria in the original sample, we need to pipette various amounts into plates. Once the dilutions are made, then plates are set up and agar is added. Plated dilution formula. So we need to figure out how much is the dilution in the plate. It's the volume plated, pipetted, times the dilution bottle. So for example, if we pipetted 1 milliliter into our 10 to the negative 2 bottle, which is what we see in our picture, our plated dilution, the amount in the plate, is 10 to the negative 2. So we can see here that we have our dilution bottles 10 to the negative 2, and we pipetted 1 mil. 1 times 10 to the negative 2 is still 10 to the negative 2. Another example is if we plated 0.1 mil. So we'd pipette 0.1 mil, we'd draw that up into the pipette, and we'd take it out of the same bottle. So now we have 0.1 mil. Recall that 0.1 mil is the same as 10 to the negative 1. Again, when we multiply exponents, we add them. So 10 to the negative 1 times 10 to the negative 2, so 10 to the negative 1 is from the pipette, 10 to the negative 2 is from the bottle, gives us our plated dilution at 10 to the negative 3. So which of these plates do you predict would have more colonies after incubation? 10 to the negative 2 or 10 to the negative 3? Correct. 10 to the negative 2 would have more colonies because it's the least dilute. Dilutions, page 222, plated dilution practice problems. Question 7 on page 222. If the volume plated, so our pipetted, is 1 milliliter and the dilution bottle is 10 to the negative 6, then what is the plated dilution? So we'll use our same formula and get our answer. So 1, which is the volume plated, or pipetted, times 10 to the negative 6 is still 10 to the negative 6. So that's how much is in our plate. Question 8. If the volume plated is 0.1 and the dilution bottle is 10 to the negative 6, then what is the plated dilution? So your equation is the volume plated times the dilution bottle. So the answer is 10 to the negative 7. So let's see how we got that. 0.1, which is the volume plated, times 10 to the negative 6, the amount in the bottle, gives us 10 to the negative 7. Or if you want to see how the math worked out, 10 to the negative 1, which is the same as 0.1 mil, times 10 to the negative 6 gives us 10 to the negative 7. So we add exponents. So now that we have plates, we need to actually count the amount on the plate after incubation. We need to look at what's called the dilution factor. So when we did dilutions, that was a fraction of the original. The dilution factor is used to calculate the original cell concentration on the next page. The dilution factor formula is 1 over the plated dilution. So it's the reciprocal or the inverse. Here's an easy way to remember that. Note all dilutions are negative exponents. They are fractions because they're a portion of the original. So if your plated dilution was 10 to the negative 7, then our dilution factors are always positive exponents because they are the reciprocal or the opposite. So if your plated dilution was 10 to the negative 7, then your dilution factor is 10 to the 7. That is now going to be a whole number when we calculate our original cell concentration on the next page. Dilution factor practice, page 223. Questions 9 through 11 on page 223. The plated dilution is 10 to the negative 2. So what is the dilution factor? Remember, it's the reciprocal or inverse, 1 over. The dilution factor is 10 to the 2, which is a whole number. The plated dilution is 10 to the negative 3. So what is the dilution factor? 10 to the 3rd. The plated dilution is 10 to the negative 4. So what is the dilution factor? 10 to the 4. So we're going to use our dilution factor, which is a whole number, to calculate our original cell concentration, which is going to tell us how much was in the original sample, in this case of E. coli. So the original cell concentration, page 224. So we started off with a broth, in this case of E. coli. We did a serious dilution using dilution bottles. We then plated those, added agar, allowed them to grow, and then we're going to see colonies. Recall the goal of dilutions is to determine the amount of bacteria cells in the original test tube. This is the original cell concentration. Original meaning the initial, cell meaning how much, and concentration also meaning how much. The units for this are cells per milliliter. So what is the formula for the original cell concentration? OCC, which is the original cell concentration, equals the number of colonies, you see those on the plate, you count those on the plate, times the dilution factor. Remember, the dilution factor is a positive exponent. It is a whole number. So the number of colonies are counted on the plate. So it's now been 48 hours, and we have colonies on a plate after incubation. Recall that only plates between 30 and 300 are used to calculate the original cell concentration. Less than 30 is not statistically valid, and more than 300 is difficult to count. So let's put this all together. So this topic is very confusing, so let's kind of backtrack and see where we started and where we're going. We started with the test tube of E. coli. We knew that it had lots of bacteria growing in it because it was very cloudy. So we wanted to dilute it to get to a number that we could actually count colonies on a plate. we pipetted 1 milliliter of our E. coli sample into our water bottle. The water bottle contained 99 mils of water. So our dilution in our water bottle was 10 to the negative 2. We got our 10 to the negative 2 from the volume added, 1 milliliter, divided by the total, 1 plus 99, which is 1 over 100. The first bottle is 10 to the negative 2. 1 over 100 is the same as 10 to the negative 2. We then diluted it further into a second bottle. 1 more mil was pipetted into a second bottle. That second bottle also has 1 over 100 for a total of 10 to the negative 4, because 10 to the negative 2 times 10 to the negative 2 is going to give us 10 to the negative 4. We then took the amounts in our bottles and we plated them. We used a pipette to plate either 1 mil or 0.1 mil in the following plates. In the first plate, our dilution in the plate was 10 to the negative 2. That is because 10 to the negative 2 times 1 is 10 to the negative 2. Our second plate, we withdrew or pipetted 0.1 mil. So 0.1 mil times 10 to the negative 2 is 10 to the negative 3. Our second bottle, we also made 2 plates. The first plate, we withdrew 1 mil. So 1 mil times 10 to the negative 4 is 10 to the negative 4. The second one, we pipetted 0.1, so 10 to the negative 4 times 0.1 is 10 to the negative 5. Lastly, we added agar to the plates, and this is the result. Our 10 to the negative 2 plate, which is not dilute, yielded 578 colonies. Lots and lots of growth, too many to count. Our second plate had of 10 to the negative 3, the 315 colonies, again, too many to count. Our third plate had 72 colonies, and it was 10 to the negative 4. And our last plate had 6 colonies, and it was the most dilute at 10 to the negative 5. So now we can take our colonies and calculate how many cells were in our original test tube to start off with up here with our E. coli. Original cell concentration practice. Question 12 on page 224. After 48 hours of incubation, 72 colonies grew on a plate. The plated dilution was 10 to the negative 2. What is the original cell concentration? So the first thing you need to ask yourself, what is the formula? Again, those 4 formulas on that previous slide is crucial. You need to know the formula. So you need to use the OCC formula. So the answer is OCC is the number of colonies times the dilution factor. Our OCC is going to be the number of colonies. In this case, it's 72 colonies times the dilution factor. Recall, the dilution factor is the reciprocal of the dilution. So OCC is 72 times 10 to the 2. The 10 to the 2 is the inverse of the dilution, which was 10 to the negative 2. Our number is not in scientific notation, so we need to continue. So now we take our 7.2 times 10 to the 3rd as our final answer, and I wrote out so you could see where that number came from. So 72 times 10 to the 2nd is not in scientific notation. We need to move the decimal 1 place. So 2 plus 1 is 3. We put the decimal between the 7 and the 2. Our units are cells per milliliter. And since we move the decimal 1 place, it's a whole number. So that's 7.2 times 10 to the 3rd. We're going to try one more. Page 224, question 13. After 48 hours of incubation, 140 cells grew on a plate. The plated dilution was 10 to the negative 4. What is the OCC? So take a moment to see if you can answer the question. So you start with the formula. Number of cells times the dilution factor. How many cells? 140. Dilution factor, how do you figure that? It's the inverse or reciprocal of the dilution. So it becomes 140 times 10 to the fourth. The 10 to the fourth was the dilution factor because our dilution was 10 to the negative 4. Next, we need to put our number in scientific notation. So how many places do we need to move the decimal to get it in scientific notation? We're always going to put the decimal between the first two whole numbers. So we're going to move the decimal 2 places. So 2 plus 4 is 6. So 1.4 times 10 to the 6th cells per milliliter is our final answer. You must always put your answers in scientific notation and include units. So here's some additional resources for practicing all of the exercises at the end of this tutorial. You also should watch the pre-lab in your lab manual because it's worth points.